Right, hi, my name is David Haak, and I'm going to talk about crosstalk and plant defenses. So plants are sessile, they can't move, so they have to integrate their responses to environmental conditions that happen to them. One of the ways that they do that is through stress pathway interactions. So here we have a herbivore feeding on a leaf that elicits a signaling molecule, genomic acid, that has some downstream target that induces a defense cascade. Similarly, under drought stress, plants produce a signaling molecule, abscisic acid, that also has some downstream target that elicits a drought response. When co-stressed, we know that these two pathways share common downstream targets, and sometimes they interact, and often the consequence of that is something like the attenuation of a defense cascade. Okay, so that's the theory, but what happens in real life? Well, we can actually go to a nice model system for that, the wild tomato. Tomato is a system where genomic acid and inducible defenses were first described. And here I've gone and collected two accessions, two different populations of Solanum pimpernellifolium. And I can grow them under common conditions and expose them to different stresses. Here, what I'm showing you is resistance on the y-axis, which is 1 minus the relative growth rate of a caterpillar feeding on leaves. In red, I've treated the plants with exogenous genomic acid, which mimics the induced defense response. What you can see along the x-axis are two populations. For LA1269, we see a nice, strong induced defense response. The caterpillars are feeding less, they're growing less. And for 1589, you see jasmonate insensitivity, no change. Well, when I co-stress in LA1269, we see that complete attenuation of the defense response. So defense is not elevated under drought stress. In our jasmonate insensitive population, we see no change, as would be predicted. So this sort of natural variation gives us the power to go in and dissect what's happening within these pathways, to identify these common targets. But how do we do that? Well, my colleagues and I decided, well, just imagine what I just said on this picture. No. So my colleagues and I decided the best way to do that is to take an RNA-seq approach. Right? An RNA-seq is the so-called digital gene expression approach, where we can stack up our reads and count differences in expression. We're doing that, but we're doing it across all treatment combinations. So we have an unmolested control, a methyljadetonate treatment, a withholding water treatment, and then the co-stress and methyljadetonate drought stress. But we're doing it across two different tissue types, so we can look at correlated responses. And we're doing it across multiple genotypes. And today I'm just going to show you preliminary data from sort of the first run in that LA1269 population. So as anybody who's sort of done some RNA-seq work, what you know is the first thing you get are thousands of genes that are differentially expressed. Right? So here I have just two replications across our multiple transitions. So which of these genes are important? Are treatments even segregating? All these things we have to answer. We can answer the latter question by taking the top 100 differentially expressed genes and using MDS to plot them. What you see is we have fairly good separation, except right here, and this is of course why we replicate. When we do this, we run this through Limavoom, one of the most popular ones. We get lots of differentially expressed genes, but most importantly what you can see is by using the combinatorial treatment, we can actually narrow that pool down pretty quickly. Now I'll save you the suspense here and let you know that of those 27 genes, almost 3/4 of them are not annotated, so there's a little bit of work to be done. The rest of them are things that we might predict. So we're doing this across multiple tissue types to look for correlated responses. And we're also investigating parallel evolution by looking across multiple species. So I showed you this polymorphism between two different populations in Solanum pimpernellifolium, but I have an entire species where every population I've looked in, there is no genomic acid response. So by going across the major groups within the tree, multiple accessions within each species, we're able to develop a better understanding of parallel evolution in this system. Whoa, the lights came out. Thank you.